Okay, it looks like most of the people have gotten into the conference room, so I'll go ahead and kick things off. Thanks everyone for showing up again today. We're really excited to have a different take and a different presentation about something that is a very important aspect of how we conserve and plan our marine conservation. Eric Treml will be giving our talk today, but before we get to Eric Treml, I'd like to introduce Ian Caldwell, who's somebody who's working tirelessly behind the scenes to line a lot of different speakers up and doesn't get a lot of credit. So Ian, if you would like to say hello to everyone and introduce Eric, that would be great. Then we'll kick off. Eric will give his talk, and then I will help moderate at the end. At the end, if you have any questions, we'll allow you to unmute yourself and ask the questions. And while Eric is talking, if you have questions that come up and you would like myself or one of the other moderators to ask Eric those questions, please feel free to write them in the chat box. So Ian, take it away.
Great, thanks, Brock. Yeah, so it is my pleasure today to introduce Dr. Eric Treml as our seminar speaker. I first heard about Eric's work when I was studying landscape ecology as a PhD student. And even then, his papers were highlighted as great illustrations of how to apply landscape ecology theories and approaches to marine ecosystems. So at that time, Eric was a postdoctoral research fellow at the University of Queensland, and he was already pushing the field of seascape ecology forward to give us a better understanding of broad-scale marine population connectivity. He is now a senior lecturer at Deakin University in Melbourne, where his Marine Spatial Ecology and Conservation Lab group focuses on the causes and consequences of population connectivity. Today, he'll be telling us about his integrative approach to biophysical modeling of marine larval dispersal, and will provide us with several examples of how his spatially explicit models can lead to more informed conservation and management decisions. So please join me in virtually welcoming Dr. Treml.
Thanks for that. Thanks, Ian. That's a very kind introduction. I hope the presentation lives up to that. Thanks for everybody for joining. It's quite a pleasure and an honor to be giving a seminar at the center. What I'd like to do over the next maybe 35 minutes or so is just give you a broad overview of a lot of the research coming out of my lab. Down here in the corner, I highlight some of our current and recent funders, but I should mention that the vast majority of what I'll be presenting is that from some really bright students, honor students, master students, and a postdoc. So I'm going to give you a series of vignettes, we'll say, kind of a meander through our research program. But all that research really falls under three key research themes. The first is asking questions about the biological and physical drivers of population connectivity. The second is how that connectivity influences biodiversity patterns. And this is biodiversity at a population, community, and even a genetic level. And the vast majority of our research is directed towards meaningful management or conservation outcomes. So this really highlights the diversity of our research. And this is the same structure that I'll use for this presentation. Again, highlighting a couple papers under each theme. So whenever I start a presentation like this, I find it important to define connectivity. It's one of those words I love and hate. You ask 10 people for their definition of connectivity, and you'll often come up with 10 different definitions. So when I think about connectivity, I think about the potential dispersal of marine organisms among habitat patches. As you all well know, that incorporates the benthic adult stage, where spawning occurs, and the pelagic larval stage. But what I often encourage people to think about is the spatial temporal spectrum over which population connectivity influences. Typically, I find myself thinking about ecological connectivity, metapopulation dynamics, recovery following storms, but more recently, getting to know some geneticists and thinking about evolutionary questions, I think arguably some ecological processes can have drastic and important implications for evolution and vice versa. So I like to kind of meander up and down this spatial temporal continuum when I think about connectivity. I think connectivity is really important for reasons listed here. The flow of organisms between population are important for population persistence, rescue following disturbances like bleaching, crown of thorns, or shipwrecks. And this connectivity or movement between subpopulations drives a lot of the metapopulation dynamics that we see in nature. Certainly, if you look at any local, regional, national, or international literature or policy framework, thinking about conservation and protection, connectivity plays a prominent role. Of course, it's not the only role, but it's highlighted often as a key or critical design principle. Thinking about where are the upstream and downstream linkages for areas of interest, for example. But of course, in practice, thinking about and quantifying connectivity is difficult. The dispersals are often quite small, underdeveloped, and dominated. The connectivity outcomes are largely dominated by strong environmental flows or currents. This leading to strong asymmetries in connectivity. And I still love this paper from, I guess, over a decade now from Jeff Jones, highlighting the diversity of tools we collectively as a community use to investigate this idea of connectivity, ranging from kind of tank and field-based behavioral experiments, some powerful otolith microchemistry approaches, all the way through population and phylogenetic type approaches. I find myself most often fitting in this, I don't know what color that is, chartreuse, purple, this biophysical modeling bubble. And I would suggest since 2009, a lot of these bubbles have expanded somewhat to cover more spatial and temporal scales. But it's difficult, and you need a diversity of approaches to ask these broad scale questions about connectivity. So at the heart of this is ocean physics. I love this movie. If you've seen any of my presentations in the past, I always put it in. I should really upgrade this video, but it really gives you a sense for the spatial temporal dynamics of our oceans. The colors here is temperature, and you can see the dynamics in, say, the Tasman front and some of the equatorial currents. So capturing and understanding these physical dynamics is critical. There's some great work out of University of Western Australia of a brand new hydrodynamic model for Australia, where they've discovered largely some new currents around the coastal zone of Australia. And of course, there's a lot of other work. People up near you, CSRO, Ames, et cetera, have some local scale models trying to quantify ocean physics at these fine to broad scales. Again, this is at the heart of the question of connectivity. So I'll have just 2 slides on my methodology, and then I'll get to some of the papers using these methods. So inevitably, all of the research in our lab starts with empirical data on behavior, mortality, survival. We use a spatially explicit approach, like Ian mentioned. We develop maps or a seascape of our area of interest, either work with hydrodynamic modelers and oceanographers, or contract this work to be developed for particular seascapes. We combine those together in various larval dispersal models, and this is kind of a classic biophysical Lagrangian-based model where we release virtual particles into this 3-dimensional hydrodynamic seascape and watch where they go, and then let them or allow them to settle on downstream habitat patches. What's nice about this numerical framework is we can do this over hundreds of thousands of times to explore different dynamics and different questions. The outcome of a lot of this work is a connectivity matrix. This might be a migration matrix or a probability matrix, nuances which I prefer not to get into at this stage, but a numerical framework for quantifying the connectivity between habitat patches. We can then interrogate that matrix and identify those life histories which drive some of the dynamics in our seascape, as well as to identify some of the important patches in a given seascape. Again, thinking about those conservation outcomes. We'll often take this connectivity matrix and push it into a metapopulation framework to ask questions about long-term survival of individual patches and implications of harvesting at individual patches, and then helping local and regional management authorities make more informed decisions. That's a Lagrangian pipeline. And this is just one slide to say that another framework that we use is using an effective transport algorithm, where we use the same type of physical and biological parameters, but we model the dispersal kernel or the probability density surface per se. So this is an illustration or a movie of individual dispersal kernels or probabilities of larvae moving between different patches in the seascape. So those are the broad scale methods. We've applied this to various regions at various different scales. Each one of these red boxes highlights a different study in our lab group with collaborators. Today, what I'm going to do is I'm going to highlight some work from this broad scale Indo-West Pacific area, some work from Micronesia, and some work from Port Phillip Bay. So I'm just going to highlight a couple papers or vignettes underneath each one of these themes, and I'll start with the biophysical kind of pillar in our research group, which asks, what are the important biological and physical drivers of connectivity? For this first paper, what we've done is we've pulled apart dispersal and connectivity into 4 major components: spawning, the actual transport, the post-transport settlement, and then the recruitment in the adult population. And of course, each one of these phases has different kind of biological and physical drivers. So what we've done is we've tried to pull in the majority of these extrinsic and intrinsic drivers into our biophysical model of Port Phillip Bay to ask the question, which one of these physical and biological drivers influence connectivity? So this work was done in Port Phillip Bay in large part with Professor Steve Swear and his lab group have been working in Port Phillip Bay for over a decade. And when we think about Port Phillip Bay and metapopulation dynamics and connectivity, we often find ourselves thinking about hula fish, right? Who wouldn't? Steve's been thinking about hula fish for a number of years and have been out sampling recruits and hatchlings throughout the bay at quite a few different sites over quite a few different seasons. So it's a beautiful, well-validated system to ask some of these questions. And working with Carrie Black, we have a high resolution, 400-meter, 3-dimensional model of Port Phillip Bay, where we're able to release virtual larvae of hula fish or urchins or kelp to explore connectivity dynamics. So what I'd like to do from this first paper is just present 2 figures. And let me just walk through the structure of these 2 figures first. So I've pulled apart the three different phases of dispersal along the x-axis: spawning, transport, and settlement, each with a suite of biological parameters. And then on the y-axis, I'm plotting the relative influence of individual parameters. So some of the outcomes might look like this, where you have a cluster of lines on a map and a bar showing the overall mean influence. So what I've done is I've done this for multiple different reefs across multiple different spawning seasons. And I've spread reefs, or that spatial signature, horizontally. So each vertical bar represents a different location in Port Phillip Bay. Spawning at different times are illustrated by colors. So if you see a difference in colors running vertically, that means there's a different influence for this particular parameter through time. So it's through time and through space. So visually looking at this plot, this large, or this parameter on the left, it doesn't matter where you're spawning or when you spawn, there is generally a very high influence of competency on the downstream connectivity. Whereas this behavior 2 parameter, the importance or the influence of that parameter depends on where you spawn and when you're spawning. So the spatial temporal context is really important. So for downstream connectivity, some of the results for Port Phillip Bay look like this. Across the board, the larval mortality, the larval pelagic duration, and the time it takes to reach competency are the main influencing factors driving downstream connectivity in the system. And it doesn't matter when you're spawning or where you spawn, these 3 parameters are consistently more influential than all the others. If we think about local retention, on the other hand, and local retention is of those larvae that are spawned from a given area, how much of those return to that natal habitat. Just looking at this figure, you can see that there are a couple parameters that are generally important, like larval mortality, that competency parameter, and larval duration. But the relative influence for a given population, it depends on where they're spawned and when they spawn. So that wide variability that you see across this parameter space shows that for local retention, determining the influence of these individual parameters on local retention completely depends on where and when you're spawning, which is a frustrating but interesting outcome. So taking this one more step further and reflecting back on the hula fish, we had a PhD student, Emily Fobert, spend quite a bit of time in Port Phillip Bay searching down hula fish, and for ages, we've had students out there pulling plankton nets in Port Phillip Bay. We haven't been able to find larvae. Emily was out over multiple sites over 24-hour periods and started to find larvae, but they were all clustered down deep within Port Phillip Bay. The vast majority of larvae that she found were deeper than 6 meters. So we were thinking this is kind of counter to the classic vertical migration of larval fish, inverts as well. And we wanted to ask questions about metapopulation dynamics for this somewhat unique system. So she did a lot of plankton tows, quantified through morphometrics, an index of larval quality, and found that the highest quality larvae are found deeper in the water column, which was really interesting. So there's a partitioning of larval quality in the water column. If we put that into our larval dispersal model, we also found that high quality larvae travel shorter distances. That is, high quality larvae stay closer to home, and it's the poor quality larvae that get dispersed to neighboring subpopulations. For us, that was a pretty profound outcome. That is, metapopulation dynamics, or connectivity in general, might be driven in large part to poor quality larvae. I don't know how applicable this is to other systems, but a really interesting outcome nonetheless. And this is just to say that we've explored different life history parameters on dispersal outcomes for a variety of taxa in this system. And this just highlights kind of our first empirical representation of fall velocity or buoyancy in larval sea urchins, how they shift from being positively buoyant to strongly negatively buoyant over this 8-day dispersal period. Taking a slightly different approach to the biophysics of connectivity, in addition to larvae, we've also worked with colleagues up in the Arafura Sea, this is Kiki Dithmers, asking questions about dispersal of ghost nets. This is a vignette that I don't want to spend a whole lot of time on, but I do want to mention this because I find it incredibly cool. So with Kiki, what we've done is we released virtual ghost nets throughout the Arafura seascape over multiple years and let them drift for up to 2 years and try to quantify accumulation and retention zones of ghost nets. And then we're eventually going to put this in context with the turtle migration routes. So we have 2 different release times shown here, where the hot colors are high densities of virtual nets and cooler colors are lower densities. And we allow those nets to accumulate on shore, highlighting net accumulation rates for different parts of the seascape. Again, there's a fair bit of empirical data from the sea rangers up in the seascape, quantifying the density of ghost nets in this area. We're writing that up right now. Okay, so that's enough about the biophysics. What I'd like to do now is take a shift and think about the biodiversity implications of connectivity. So a lot of this work is inspired by papers I've read by Briggs and Bowen and Kent Carpenter and Paul Barber and others, thinking about broad scale biogeography, and just reflecting on kind of some of the maps I was looking at and trying to figure out what role does dispersal have in developing these broad either biogeographic provinces or regionalization, or dispersal barriers between between islands or among sea states. So I'm thinking about like the Wallace line or the marine Wallace line and things like that. So what we did is we developed a model for the Indo-Pacific. We modeled dispersal for over 100 species. And then we sampled this virtual seascape at different research stations. So each one of these points is a real research station or a real sampling point from some of our colleagues. And I simply wanted to ask, what's the likelihood that a given life history ensemble or a virtual species would have the capacity to disperse and move between sample sites? Again, thinking about biogeography and phylogeography. So we used our advective transport algorithm to model many different species, tessellated the surface, developing these boundaries between every pair of sample points, and simply asked, what's the proportion of species that can or cannot make it between sample sites? So the lines that you see on this figure represent the proportion of taxa that could not make it between sites. So these deep heavy lines, as you would suspect, crossing into Micronesia or to Palau from the Philippines, most species cannot make that dispersal event based on their life histories or the physics. So we have this, for this broad scale, I guess, representation of dispersal barriers, which I thought was interesting. Others perhaps thought it was less interesting. But then I thought about, what are the biological or physical drivers of these patterns? So for each one of these tessellated edges, we used a logistic regression to ask what's the influence of reproductive output, larval duration, mortality on the ability to pass over that barrier. And as you might suspect, as your reproductive output increases, you have a greater likelihood to pass a barrier. Nothing surprising there. As you're able to survive longer in the plankton, the greater capacity is to pass some of these barriers. Same thing with mortality, increased mortality, reduced likelihood. But what I found really interesting, again, is this temporal signature that it completely depends on when you're spawning. The spawning periodicity, the time of spawning dictates the likelihood to breach, to make it past some of these dispersal barriers. So again, it's the impact of spawning phenology on dispersal outcomes, which I find really interesting. So I had this opportunity to work with a large group of really bright geneticists and modelers, and this is the Diversity of the Indo-Pacific Network, DIPNet. You can Google that and check out the website. But this is an opportunity where a bunch of different collaborators put all of their georeferenced sequence data online into a database. So in this database, we have over 56 taxa, 35,000 sequences for 1,100 different locations. And we thought it was a brilliant opportunity to ask questions about the phylogeography of multiple species over a broad seascape. This first map shows the density of species sampled, as well as the number of sequences. It is a lot of data. Moving into this project a couple of years ago, we thought this is a beautiful opportunity to finally come up with some great empirical-based validations of some of these bioregionalizations. As you know, there's a number of bioregionalizations published. There's 8 of them here. So we tested how well our genetic data, as in DIPNet's genetic data, fit or did not fit this bioregionalization. The outcome was somewhat underwhelming. There was some support for broad scale regionalization. So some of the barriers that we saw in the regionalizations with 5, 4, or 3 divisions had the most support, but only 40% of the taxa supported those findings. So the majority of the taxa, there was no effect, which was a real disappointment. Left us really scratching our head and pulling out our hair. Why could this be? Perhaps the mitochondrial DNA wasn't well suited. Maybe we're faced with a non-equilibrium situation. Maybe historical processes play a more prominent role than dispersal of the current environment. Or there might be some nuances with respect to the amount of dispersal and whether it's ecologically or evolutionarily significant. We're still working on this. This is a great effort, is very enjoyable, but this end result was, I guess, somewhat frustrating. Watch this space. There's a lot of people working in this group on some future papers. So another related question in terms of the biodiversity implications of connectivity is testing hypotheses of isolation by dispersal distance, or the analogous to IBD, isolation by distance. So this is working with Sarah Davies, who at this time was at University of Texas, now at Boston University. And she went out and sampled 2 different corals from, I think, 10 different sites in Micronesia. So you had a great empirical based data set. And we wanted to ask a question about the role of connectivity in this system. So what she was able to do is she looked at the relationship between pairwise genetic distance and modeled biophysical modeling distance for 2 different species of Acropora. We did this for 2 different larval durations, because there was some uncertainty on what that larval duration parameter should be. So we modeled this for a 90-day larval duration and a 65-day larval duration. What I thought was really cool about Sarah's work is that she was able to lend support for a life history characteristic based on the fit between the genetic data and the model data. So here we see that for Acropora digitifera, there was a better fit for this 90-day larval duration, whereas for the other coral, there's a better fit for the 65-day larval duration. So again, using a combination of methods, we're able to kind of back out the some of the unknown life histories, or at least lend support for some of those life histories, which I thought was really cool. So in a similar way, where we use a genetic biophysical modeling framework to learn about life histories, this is work that was completed by Niels Krook from University of Queensland at the time. He's now down at University of Tasmania. And he was interested in the sea mullet and asking the question, we don't really know where sea mullets spawn. Are they near shore, offshore? Are they 50 or 100 kilometers north of the estuary site? So what Niels did is he collected a lot of data from a lot of different sites, and we modeled the larval connectivity as these fish swim upstream or downstream and spawn. Niels tested 12 different locations of spawning, but he went a step further and he modeled genetic differentiation on our migration matrices to come up with a modeled FST, and compared that with his observed FST from his SNP data. And by using this structure, he was able to lend good support for the model that suggested that the sea mullet spawns 50 to 100 kilometers upstream from the estuaries and near shore, not offshore or on the slope area. So again, a really cool application integrating biophysics with genetics to learn more about the life histories. So at a slightly deeper, I guess, time scale, we've been thinking about what role could migration and connectivity play in the local adaptation of species, thinking about climate change. So we've had some early maps, this from a master's student of mine, looking at connectivity of an acroporid in contemporary time periods and in 2100 from some future CSRO modeling. We're asking questions about what drives those differences in connectivity, but working largely with Misha Matt from University of Texas. What we've done is we, as in Misha, has pulled our dispersal models together with some metapopulation models and individual-based genetic models to model gene flow. So now we're not only modeling larvae, but we're modeling the genes on those larvae and tracking if they come from a heat-tolerant or a heat-sensitive population. So in large part, asking kind of what-if questions. What's nice is Misha has the numerical and the genetic background and the modeling background to kind of stitch all of this together. He's been sampling in the GBR for a while and with local colleagues as well, Madeline Van Appen and others. And we're able to compare our genetic networks, with the biophysical networks, and come up with a nice fit. And then we're able to look at future implications of warming across this broader seascape. So we've modeled warming under different climate change scenarios with migration in a demographic model, and tracking hard and soft selection and how that changes the allele frequencies at downstream sites. Making a very long, complex story short, some of the outcomes that Misha has found is that future populations perhaps will be more sensitive to thermal fluctuations, just based on those end of simulation allele frequencies. Perhaps the standing genetic variation may be enough to fuel rapid adaptation, but again, that's context dependent. And some regions across the seascape show higher adaptive capacity than others. I love this, this map from Misha's recent paper showing the percent coral cover and how it changes 100 years from now and 200 years from now. And all of this kind of central Indo-Pacific seascape is effectively devoid of reef cover. Going a step further, these papers suggest that local scale population level persistence may be driven in large part by the proportional gene flow from warm adapted sources. So if you have a warm adapted population feeding larvae into a downstream population, that will increase the persistence capacity of that downstream population. And another important parameter is the present day thermal environment, the mean and variability in temperature. All right, so with that broad scale and admittedly very quick meander through some research projects we've been working on in the world of biodiversity, I'm going to take the last couple of minutes and reflect on how some of this work is influencing management. So some of our earlier work tried to map multi-species dispersal corridors. Kind of one of the holy grails, I think, of marine conservation and management is identifying management approaches that work for multiple species. This is one of our first representations of multi-species dispersal. I was pretty excited about that. Bringing this to some of the broad scale managers in the Philippines. At this time is with the Coral Triangle Support Partnership, the CTI initiative. A lot of those interactions with managers were somewhat underwhelming, simply because, as you perhaps know, managing seascapes at local and regional scales does not happen on a reef by reef basis. It happens between management entities, provinces, states, federal authorities, things like this. So what we did is we took this ecological connectivity on a reef by reef basis and cookie cuttered that with kind of a broad scale management regime. And here we illustrate this with Spalding's ecoregions. So now instead of looking at the connectivity between coral reefs, we look at the connectivity between management areas, effectively identifying those upstream and downstream ecological neighbors of management units. This was done at this broad scale, but we're also looking at some fine scale work both in the Philippines and in Indonesia. So making connectivity, making ecological connectivity a little bit more appropriately appropriate for management. So I have just a couple slides looking at some of our work with an NGO called RARE. And this group is interested in working with locals in developing seascapes to better sustain subsistence fishing. So it's bringing scientists together with local communities to get some good conservation and management outcomes, perhaps not necessarily for biodiversity, perhaps more for fishing persistence for many generations. And this is just an example from Southeast Sulawesi. And a lot of this work, the vast majority of this work has been done by my postdoc, Kay Critchell. So the pipeline for RARE starts by identifying key areas of interest shown on this map by little polygons. These are communities that are keen to develop protection to help build a sustainable fishery for the local communities. We take some of that local information, develop a hydrodynamic model with colleagues, do some more of this larval dispersal modeling, and come up with reef specific conservation value or management value. So for each node, we're able to say that this node is important to your fishing ground or it's less important. And they can take this with a bunch of other information to make their management decisions. So they can take this information and others through some community-based consultations, determine what their system, their protected system requirements might be, identify priority areas, and then implement those management outcomes. So actually designate protection areas. This is some work that that I really enjoy because it kind of brings all of this science to some very tangible, real management outcomes in the field. My last two slides are just taking a sidestep. Up until this point, I've been talking about connectivity as being a benefit. Increased connectivity builds resilience, recovery following disturbances. But in different systems, connectivity is less good, or rather it's harmful. And this is a system that we've been working in, again, with one of our PhD students, Fran. She's now down at CSRO in Tasmania. But this is looking at sea lice movement between salmon farms in Norway. The idea is that we wanted to model sea lice movement, infection rates, very similar to metapopulation dynamics and reef systems. But the main goal here is how to break the system, how to break the flow of sea lice. So through some of her work, she's used network analysis to come up with the seasonal patterns of sea lice movement. On the left here, this is summer, and this is winter. And this is kind of the natural clustering of farms across these seasons. So Norway has actually taken up this information, and they use this grouping to manage the farms in their seascape. Her last paper was helping to identify fire breaks. So if the Norwegian government wanted to set up a fallow system where they shut down farms to minimize sea lice movement, where would those be? And using a very similar framework of modeling sea lice dispersal, network analysis, and attacking that network to identify those key nodes that will break up that sea lice network. She's able to identify some key areas where removing a subset of farms could drastically reduce flow of sea lice throughout the seascape. Again, combining biophysics with a validated field-based system to come up with real management outcomes. All right, so I think that was 35 minutes or so. Hopefully some of that made sense. Again, walking through those three kind of research themes of understanding the biophysics, implications for biodiversity, and how that might influence management. If that was a little bit too fast, I'm happy to leave a PDF of this presentation with Ian, and you can feel free to distribute that. I'd like to thank all of our funders, colleagues, and thank you for attending. And with that, I'm happy to take any questions.